All right, welcome back everyone. So most of this video we're going to talk about membrane transport. Membrane transport, as the name suggests, is how things move across the plasma membrane. Whether that molecule is going into the cell or out of the cell, there's a whole wide range of mechanisms by which that molecule moves. And these are all the different mechanisms. So we're talking about kind of what this drawing shows us. How does a molecule go into a cell? How does a molecule go out of a cell? What determines whether that molecule goes in or out is, what kind of membrane proteins are there? What is the molecule? What's its chemistry? So, for example, if you are small and non-polar, you can usually go right through the membrane without a protein. If you're a larger molecule or if you're a polar molecule, you're going to need some sort of membrane protein. And then it also has to do with what we call a concentration gradient. Things want to move from where they're in a high concentration to where they're in a low concentration. All right, that's called an ion or concentration gradient. So that also plays a big role. So looking at this figure, you'll notice this red line here. So that red line separates these mechanisms that don't need ATP from these mechanisms over here that do require energy in the form of ATP. So you'll notice over here, if you don't need energy, we call that a passive transport. All right. If you need energy, you're an active process or active transport. So let's look at the passive transport mechanisms. One method is simple diffusion. So notice simple diffusion does not need a membrane transporter. All right, so you don't need a protein. So it's kind of like what's going on here with this arrow. So some things, some molecules, can go right through the membrane without needing a protein. All right, those are, again, usually going to be small, non-polar molecules. Okay, and the direction is determined by the concentration difference. So if there's a lot of the molecule on this side and very little of it on the inside, that molecule is going to go from where there's a high concentration to where there's a low concentration. Okay? Osmosis. Osmosis is a form of simple diffusion, but it's water. Okay, so osmosis is diffusion. It's diffusion of water. So now if you have a lot of water molecules here and less water molecules here, water is going to want to go through that membrane. Okay, now water is a tricky one because it will go through the membrane, but not very well. If you want it to go really well, you need what's called an aquaporin channel. We'll talk more about those as we go. All right, so that's simple diffusion and osmosis. Some things, if you can't get through just those phospholipid bilayer, you need to be facilitated. You need help. And the help comes from a protein, all right, a membrane protein. So facilitated diffusion is just like simple diffusion, except it requires some type of protein. Okay, so the molecule is going from high concentration to low, but because it can't get through the membrane on its own, it needs help, or it needs to be facilitated using either a carrier protein, a channel protein, a gated channel, a leakage channel, or if you're water, you use an aquaporin channel. Okay, and we'll talk a little more about those, but that's facilitated diffusion. All right, over here on the left side is when you need energy. So you could use a vesicle. So vesicular transport is when you use a little membrane vesicle, all right, to bring either something into the cell, endocytosis. You might bring something out of the cell, exocytosis. You could bring something through the cell. So transcytosis is when it's basically the combination of endo and exocytosis. So endo brings the molecule in, and then exo brings it out. So it can literally go through a cell, and that's called transcytosis. Vesicular trafficking, that's using vesicles to kind of move things around inside of the cell. So that's using some sort of membrane vesicle, or a little container, if you will. Primary active transport is using a protein to move a molecule through this membrane, but it's opposite diffusion in that you're going from a low concentration to a high concentration. Okay, so that's the key difference. If you're going from a low concentration to high, you need energy. But if you're going from high concentration to low, you don't need energy. All right, and we're not going to really talk about secondary active transport. That's typically when moving one molecule causes you to move a second molecule. Okay, but we're not going to really go there. All right, so this is just kind of, summarizing some of these things using, this table here. You also want to look at drawings. Drawings really help you, visualize these processes. So let's look at diffusion again. So by definition, passive movement, meaning no energy, of a molecule, could be an ion, could be, any kind of molecule, down its concentration gradient. So when we say down its concentration gradient, it means it's moving from where it's highly concentrated to where it's low concentration. So that would be, for example, these lipid-soluble molecules. All right. If this was where it's highly concentrated, notice there's, not that many more, but they're showing 5 molecules. If there's less here, that's a concentration gradient. That molecule will go through the membrane, doesn't need a transporter, doesn't need a protein, so it's known as simple diffusion. All right. Examples. The big ones are the gases. Oxygen. This could be oxygen. All right. Carbon dioxide. Those two gases cross membranes by simple diffusion. So when we get to the respiratory system and we learn that, oxygen in your lungs is going to diffuse into your blood, that means it doesn't need any transporter, doesn't need energy, doesn't need, a membrane protein. Facilitated diffusion, keep one thing in mind, that the only difference is it requires some sort of membrane protein, either a channel or a carrier. All right, so that would be shown here. So there's the molecule, high concentration, going through the channel where it's in low concentration. Okay, so this is simple diffusion. This is facilitated diffusion. All right. Osmosis, only difference there is we're dealing with water. So now instead of looking at maybe a lipid or an ion, we're looking at water. So water will go from where there's a lot of water, a high concentration of water, to where there's a low concentration of water. Another way to look at osmosis is not looking at necessarily the water concentration, but where you have a lot of solute. Solutes are everything else. Solutes are going to be like sugar or sodium, potassium, anything else besides water. If there's a high concentration of solute, water is going to travel towards that high solute. All right? Because where there's high solute concentration, there's low water concentration. And I'll kind of get into that again in a minute. So here's a couple more drawings, I guess. Simple diffusion, another example would be if you drop a crystal in water and it diffuses into that water eventually. That's diffusion. It's not through a membrane, but it's going from where there's a high concentration in that cube or crystal to where there's a low concentration, that distilled water. I like to sometimes use the analogy of putting cream in coffee, right? Why does that cream diffuse throughout your coffee? Because it's going by simple diffusion. All right. Another example of facilitated diffusion is how we bring glucose into cells. So glucose is that, really important carbohydrate that cells use for energy. Well, how do cells bring it inside? It's water soluble. It's polar. So glucose cannot go through that membrane. So in order to get glucose into a cell, the cell needs a glucose transporter. And this purple protein represents a glucose carrier protein. All right, it grabs the glucose on one side, changes its shape, and then releases the glucose into the cell. All right, back to osmosis. All right, so again, what is osmosis? It's water moving by diffusion. Okay, this is where we introduce a few more terms. All right, hypotonic, isotonic, and hypertonic. So what we want to do is look at the front part of that word. Iso means the same. Hypo means low. Hyper means high. What we're talking about is the solute concentration of the solution. So what we're looking at here are red blood cells. All right, and if you put a red blood cell into a solution, okay, that has the same concentration of solutes as the inside of the red blood cell, okay, that solution, therefore, is known as an isotonic solution. All right, so notice down here, isotonic solution, one having a concentration of solutes equal to that inside of the cell. If you put that red blood cell in an isotonic solution, water will go in and out of the cell, but equally. And that cell maintains its appropriate shape. All right, red blood cells look like this. They have these little concave surfaces on each side. That's what you want your red blood cell to look like. Okay? Now, so that one's easy, as long as you remember iso means same. Hypo means low. All right, so this solution has a low solute concentration compared to the inside of the red blood cell. So what is water going to do? Water is going to go into that cell. Okay, now remember why. Water is going to move from where water is highly concentrated, that's in the solution, to where it's lower concentrated. So again, the water here, the solution, has low solute but high water concentration. So they're basically inversely related. So when you see hypotonic, you want to think low solute, all right, but high water concentration. And therefore, water is going to go into the cell. And it could rupture that cell. So you don't want red blood cells in hypotonic solutions because they'll rupture. All right. Over here, we have hypertonic. Hypertonic means high solute. It's kind of like putting cells in salt water. If you put this cell in highly concentrated salt water, the water now, instead of going in, goes out of the cell because water is going to go towards higher solute or lower water. Okay? This is one of these concepts that students struggle with. So if you're listening to me and you're completely confused, it's okay. It's normal. But you got to get there. You got to get there at some point. Okay? So remember that when you have a solution that has low solute, that solution is going to have high water. When you have a solution that has high solute, it's going to have low water. And water is going to go towards the lower water concentration, aka the higher solute concentration. Okay, let that sink in. If it doesn't make sense, you got to listen to that little part of the lecture again. This also explains why, if say you're in the medical field, you don't give an IV of pure water. If you put pure water in someone's bloodstream, this is what's going to happen to their red blood cells. Their red blood cells are all going to explode. This is also why you don't put an IV with high salt. All right, you don't want to put a highly salt solution in someone's blood because all their cells are going to shrivel up. So when we put fluids into someone's bloodstream, that fluid needs to be isotonic to the red blood cells and white blood cells. Okay, next thing, active transport. Remember, active transport means energy. And why do you need energy? Because you're going to be moving the molecule not from high to low, but from low to high. All right? One of the analogies people like to use is like a hill. When you roll something downhill, it doesn't take any energy. All right? When you're rolling something uphill, you got to put energy. You got to put more force behind that. Okay? So when you're moving something from high to low, it's passive, no energy. When you move something low to high, you need energy. And the most important active transporter in animals is the sodium-potassium pump. All right, sodium-potassium pump is going to be extremely important for us to understand when we get into the nervous system. Basically, it uses energy to move sodium ions out of the cell. So there's the sodium ions. Now they're moving into the channel, or I should say the transporter, and then they move out of the cell. At the same time it's moving 3 sodiums out, it brings in 2 potassiums. So that's why it's called the sodium-potassium pump. But notice the ratio. It's 3 sodiums for every 2 potassiums. Okay? Extremely important, especially when we get to the nervous system. All right, here's, kind of a review of the vesicular transport mechanisms. Remember that they require energy and they're using a vesicle. A vesicle is a little membrane container inside the cell. All right, so notice we've got endo and exo. Endo means inside. So we're bringing something inside of the cell. So this is an example here, all right, where these green little molecules are entering the cell. Pinocytosis is an example. It's a type of endocytosis. Notice on the other side, all of a sudden now it's exocytosing. So the combination of the endo and the exo is transcytosis. So transcytosis is movement of a molecule through a cell. All right, endo on one side, exocytosis on the other side. All right. Phagocytosis is a type of endocytosis, but you're bringing in a large particle. All right. Pinocytosis is when you bring in small molecules. Phagocytosis is when you're bringing in something big, like a bacterium. All right. This could be a bacterium or a virus, and the cell is bringing it into the cell. All right. Now, what this doesn't show is this vesicle will fuse with a lysosome, and the enzymes will kill that bacteria. Another form of endocytosis is called receptor-mediated. Basically what that means is it uses these little receptors to grab a hold of the molecule, then it endocytoses it. All right, and an example of where this is done is when the cell brings in cholesterol. So cholesterol enters cells by receptor-mediated endocytosis. All right, so that's all the transport. That's a lot. Again, you kind of have to get through the different mechanisms. Reach out to me if any of them are giving you trouble. But once you get through those, we're about done here. So this is, you've seen this slide. We've had the same exact slide in the previous chapter. I'm just reviewing again what transcription is and what translation is. So DNA making mRNA from the DNA is called transcription. Again, it occurs in the nucleus because that's where the DNA is. Then that mRNA can be transported and finds a ribosome. The ribosome is either part of the rough ER or it's just kind of loose inside the cell. And the ribosome will then translate the mRNA and make protein. Okay? All right, finally, we're going to talk just a little bit about cellular replication. I'm not going to go into all these different phases. If you had biology, you probably had to go through all these phases of what we call the cell cycle. Basically, what that means is a cell kind of goes through this cycle in that it kind of does its thing and then eventually has to divide into two. Not going into all the details here, but I want you to know that mitosis, okay, you start with one cell and it becomes two identical cells. All right, so the two cells that are made are exactly like that first cell. All right, so they're genetically identical. So this is how you grow new skin, right? You're always producing new skin cells, so those skin cells undergo mitosis. This is how you're maybe growing, cells in your oral cavity, your mouth. Let's say you scratch yourself chewing on food and you have to replace those cells. So mitosis is how, most of your cells of your body replicate. Okay. Now, some cells can't, like a neuron, for example. A neuron loses its ability to mitose. It can no longer divide. That's why you have to be nice to your neurons. They're the only neurons you're going to have. Neurons are what we call post-mitotic or amitotic. It means they're no longer able to divide. So that's why nerve damage and nerve injury is so hard to treat, because we haven't figured out a way to take a neuron and make it divide. Now, the field of stem cells might one day help us replace nerves, but we're not quite there yet. All right, so that's the cell cycle and mitosis. Meiosis, all right, so this is kind of the home run slide here, kind of what to know about the two. So let's go over mitosis again. So what cells undergo mitosis? We call them somatic cells. Somatic cells are all your cells other than sperm and egg. All right, so somatic cells undergo mitosis. Again, neurons are kind of the exception. They can't do it anymore. How many divisions are there? There's one, because you take one cell and it becomes two. All right, so there's one round of division. Those two cells are genetically identical to each other and the original cell. Okay, how many chromosomes? Well, you have 46, right? You have two sets of 23 in a somatic cell. When it divides, you got two cells that have 46, all right? So it's taking a diploid cell that has the two sets, right, 46, and you produce two more diploid cells, all right? And this happens, again, all throughout the body. All right, so what is meiosis? Well, meiosis produces sperm and egg. So you got meiosis in a male, that results in sperm. You got meiosis in the female ovary, that results in eggs. There's two rounds of division. So you actually start with one cell, it divides into two, and then those two divide into four. And during this process, these cells are going to be genetically different. All right? That's why children are different than their parents. They're similar, but they're not identical. All right? So this allows us to have organisms that undergo sexual reproduction and produce genetically diverse offspring. Okay? So the result are haploid gametes. Okay? So haploid means these four cells aren't going to have 46 chromosomes. They're going to each have 23 chromosomes. So now if you have a sperm that's haploid that fertilizes an egg that's haploid, the result will be a diploid organism. All right, and that's called the zygote, and then that becomes an embryo, and then that becomes a fetus. All right, where does this occur? It happens in the gonads. All right, that's where meiosis occurs. All right, that's it. That's it for chapter 3. Again, hopefully this isn't totally new, but it might take a little time to refresh yourself. All right, thank you, and we'll see you in chapter 4.